Hello and welcome to the second video on energy production, mitochondria and the TCA cycle. So the purpose of this lecture is to understand how the citric acid cycle contributes to energy production. So what is the citric acid cycle? So the citric acid cycle is a series of chemical reactions that occur in the matrix of the mitochondria. You'll see in many textbooks that this is also called the tricarboxylic acid cycle or the Krebs cycle. And the reason it has multiple names, and certainly the Krebs cycle, is because it was first discovered by Hans Krebs. And he called it the citric acid cycle. So for the purpose of this video, we're going to stick with Hans Krebs' original name, the citric acid cycle. And this series of chemical reactions uses cellular metabolites such as pyruvate or fatty acids to produce high energy molecules. A waste product of this, which we all breathe out when we respire, is carbon dioxide. This Krebs cycle or citric acid cycle is also used to synthesize intermediates for amino acid synthesis. So glycolysis, which we've covered in previous videos, results in the production of a molecule called pyruvate. Pyruvate can then enter the mitochondria. Remember, glycolysis will take place in the cytosol. So pyruvate then enters the mitochondria and is converted to a molecule called acetyl-CoA. Acetyl-CoA will then combine with oxaloacetic acid, and this will then enter a cycle. Waste product, as we talked about before, is CO2. We get the production of 8 high energy electrons and 1 molecule of GTP. Remember, ATP is used for energy. Well, GTP is a very similar, obviously it's another nucleotide. The GTP will then be converted to ATP. But GTP is also important because it's involved in many signaling pathways. If we look at the citric acid cycle in more detail, so we have our acetyl-CoA at the top, the C2, so we have a 2-carbon molecule which combines with oxaloacetic acid, which is a 4-carbon, to form the 6-carbon molecule citric acid. This is then sequentially broken down from 6 carbons to 5 carbons to 4, and on its way we were producing CO2. And we're also producing these high energy carriers, NADH, on the right hand side. And on the left hand side we have GTP, which is the high energy molecule. And we have another electron carrier called FADH2. So what are these electron carriers? Well, NAD, or nicotinamide adenine dinucleotide, NAD plus is an oxidizing agent, and what this does is it accepts electrons from other molecules and becomes reduced. And these high energy electrons are then passed on to the electron transport chain, where the energy of these is used to create a proton gradient, and this we will cover in the next video. But sufficient to say at this point in time is that these carry the high energy electrons, which are derived from essentially the breakdown of glucose. Remember, glycolysis will feed the citric acid cycle, and it's this breakdown of the carbon backbone which is providing these high energy electrons. Similarly, flavin adenine dinucleotide is another electron carrier, again accepting electrons, and becomes reduced to form FADH2. And again, this molecule will provide the high energy electrons to feed the electron transport chain. So at this point I have a short multiple choice question for you. And which one of the following locations is where the citric acid takes place? Is it A, on the cristae, B, in the inner mitochondrial membrane, C, in the intermembrane space, D, in the mitochondrial matrix, or E, in the outer mitochondrial membrane? I'll just give you a second to pause the video before I reveal the answer. And of course the answer is the mitochondrial matrix. Remember that the enzymes required for this cycle are present in the mitochondrial matrix. So similar to any biochemical reaction you'll find in the body, this is controlled by a series of enzymes, and these enzymes are regulated by metabolites. And you can see clearly from this diagram the major regulators of this cycle are ATP and NADH, or ADP. And as this cycle is involved in the production of NADH, and it's also indirectly involved in the production of ATP, high levels of these two molecules will inhibit the enzymes that regulate this cycle. Conversely, if we have high levels of ADP, which means that the ATP has been used up, so we have high levels of ADP, this will promote the activity. Now, I'm not expecting you to know the enzymes. In fact, I've not even given you the names of the enzymes. This is not something, but you need to understand that this is receiving pyruvate from glycolysis. This passes into the mitochondria where it's converted to acetyl-CoA, and then it's sequentially broken down from the 6-carbon citrate through to the 4-carbon back to oxaloacetate. And in the meantime, it's producing those high energy electrons which are carried by the electron carriers NADH and FADH2. So here's a nice little overview. So in the lectures so far, we've covered glucose being converted to pyruvate by glycolysis. In today's lecture, we've learned that this pyruvate is then converted to acetyl-CoA in the mitochondria, which enters the TCA cycle, or the citric acid cycle, to produce to give electrons to the electron carriers. So the next step is the process called oxidative phosphorylation, and this is the process whereby ATP is generated using the energy from those electrons which are carried by NADH and FADH2. And this reaction requires oxygen. So this oxidative phosphorylation occurs in the mitochondria. The proteins involved in this complex, and these are big protein complexes, are present in the inner mitochondrial membrane. And it consists of 3 complexes which are passing electrons on, and each time the electrons are passed on, they release energy. A typical exam question we could ask, a multiple choice question, is which one of the following processes best describes how the high energy electron carriers that drive ATP synthesis are generated? Again, if you'd like to have a guess at this, I'll just wait a second while you pause the video. And of course the answer is the citric acid cycle. Gluconeogenesis, which we will cover in another video, is the production of new glucose from non-carbohydrate precursors. Glycolysis, we've already covered, is the breakdown of glucose. Oxidative phosphorylation is actually the process of generating the ATP, not the production of the higher energy carriers. And the pentose phosphate pathway, again, we will cover this in another video. And that concludes this video. If you'd like to do any more reading about the citric acid cycle, then this is covered in chapter 17 of Stria. Thank you all for listening.